uh, hydrocarbon backbone. Proteins. We've talked about fats and carbohydrates. Now we talk about proteins. Proteins are the most abundant molecule in your body. These are the Legos. Remember, like amino acids that make up your proteins. These are like Legos, and I can disassemble these Legos and then reassemble them into pretty much anything. There are so many things that are categorized as proteins, amino acids in your body. That's pretty much, they have the widest variety of functions. And this was the list of our amino acids. Now, I am not going to have you identify these on a test yet. When we get into nutrition, we are going to start talking about these amino acids in pretty great detail. We're going to talk about what do they do, what do they combine with, which Legos combine with which Legos to produce which Legos. And so we're going to get into this in pretty great detail when we get into nutrition. But for right now, it's just so you've seen the list. Now, the structure of proteins, we talked about this yesterday and I compared it to origami. We have some specific ways that proteins are folded and created. And it is these folds, this creation, that allows your body to know what to do with it. What job does this protein have within your body? So the first we had was our primary structure. And this is where I'm going to take my amino acids, I'm going to take my Legos, and I'm going to line them up into a chain. Secondary structure is when I allow that chain to have natural bends. It's like when you have your computer cord or an extension cord, and you go to fold it up and it follows the natural bends of the cord. That's what we're talking about with the secondary structure. Tertiary is when I now take that and now I make the very fine little origami folds, making this protein specific. And then I can take that specific molecule of protein and I can bind it with another molecule of protein, and that makes a quaternary structure. So all we're saying here, it really is just terminology, is what kind of bending, what kind of folding, what kind of manufacturing am I doing to make this specific protein? And why does this protein look different and function different from the other proteins? Here we have our little picture for you little visual learners. So here I have my chain. If I have a chain of proteins, I have made primary structure. Then I allow it to follow its natural bend, its natural kink. That's going to be our secondary. Then I go in and make my little origami folds, making this protein very, very specific and original, tertiary. And then I have here two complex proteins that I have bonded together, quaternary. We have some specific jobs for these proteins, and this is just kind of going over some specifics. We have structural proteins, and structural proteins are just that. Their job is to provide structure, to provide support to different areas of the body. One example is your collagen. Collagen is an elastic connective tissue under your skin. Now, unfortunately, as you get older, gravity works, and gravity is constantly pulling on your skin at all times. And that collagen can, in fact, lose some of its elasticity. And so what do? We can give ourselves injections, we can rub collagen on our faces and cause that structural protein to firm back up. Keratin is going to be the protein that gives our fingernails and our hair its structure. Going to give us also our horns, antlers, hooves. Actin and myosin. These are going to be two proteins that we're going to talk about a lot. And these are really interesting. All right, so let's say I have a muscle. These are the ends of the muscle. Here I have actin and myosin filaments, kind of like this. Make it a little longer. So these are actin and myosin filaments. We have a little zigzag down the middle. So this is a muscle. This is your bicep. And basically what happens with these actin and myosin filaments is they're kind of overlapping a little bit like this. But when your brain tells your muscle to go ahead and contract, they ratchet over each other, shortening towards the middle. So if I tell this muscle, I want you to shorten and I want you to contract, we can see how long it is from here all the way to here. But then if I have those proteins ratchet over, it has now shortened by about half of the length. And so we have shortened and thickened the fibers. And those are because of these structural proteins, actin and myosin. Yes, I do have it recording, Vanessa. I just saw your chat. Thank you. All right, so next we have our functional proteins. These are proteins with jobs. Always reminds me when I say that of that subreddit called Dogs with Jobs. I love that subreddit. Anyway, so some of our functional proteins can include hormones. Everything about your body is governed by hormones. How you use nutrients, which is your metabolism, how your brain learns things. Sorry, my body has been trying to sneeze literally all morning. It has just, I got a sneeze stuck in my nose and it won't come out. I've even tried looking at the light. I know I can look at the light. Yeah, well, I'm not going to burn out my retinas, but usually looking at a light works, but it just won't come out. So everything from your red blood cell production to your metabolism to your sympathetic nervous system, parasympathetic nervous system, your reproduction, everything is controlled by hormones. The color of the skin, the color of your hair, all of this, hormones. And all of these hormones are functional proteins. And it is the shape of that protein that tells your body what to do with it. The shape of the hormone that gives you skin color is a different shape than the hormone that controls your metabolism. And this is how your body identifies it. And, oh, damn it, it's not going to come out. Oh, that one made my eyes water. And it tells your body what to do with it. Antibodies, we talked about this yesterday. I find immunology really, really fascinating, especially right now that we're really dealing with immunology on a daily basis. We have these things called antibodies. And antibodies, now obviously yesterday I, simplified it to the nth degree. But what antibodies are little proteins that walk around with a wanted poster. And on that wanted poster is the picture of a foreign invader that has invaded us before. And when he finds it, he can immediately signal Immunity Groot. And Immunity Groot already knows how to get rid of that foreign invader. And so it is this antibody, this immune recognition that allows us to launch an attack against it. We have additional hormones and then enzymes. And we talked about that. Enzymes are primarily just a catalyst being something that starts a reaction. Usually these enzymes are going to start, or end in the three letters ACE. Some examples would be like amylase and lipase, which are digestive enzymes that we find in our small intestine as well as our saliva. So they're catalyzing. They are beginning and speeding up chemical reactions. All right, is everybody okay? Is everybody on the bus? Hello. Okay, the rest of you have no idea what I'm doing right now, and that's okay. One of you does know what I'm doing right now, and I'm saying hello.
Myself, I'm here. I was just late.
I got you. Done. All right. So these enzymes that are beginning and starting some sort of reaction, they're very specific. Oh my God. I might just have to go excuse myself for a second, blow my nose or something. They are very specific. Think of a lock and key. One enzyme is going to start one reaction, just like your car key starts your car. Hopefully your key doesn't start anybody else's car. That used to be a big problem back in the olden days, is that keys would start multiple cars. Now they've got it pretty well figured out. My key will only start my car. So if my key is the enzyme and my car is the reaction, this is why we call it a lock and key, because that one enzyme is going to pass by every other reaction until it fits into the one that it is supposed to initiate. All right, we started talking yesterday about nucleic acids, and this is the building blocks of things like DNA and RNA. DNA is what really makes up you. DNA is contained in the nucleus of the cell, and it is what has your genetic code. Everything about you was pre-planned. You are a foregone conclusion. I hate to say it. You also, this is really kind of fascinating though, have little bits of DNA. When I say DNA, I'm talking about repeats. So remember we talked yesterday where we had, oops, here's my nucleotides, my nucleic acids. So there's A, T, G, C, G, A, G. If we repeat these in any way, depending on the type of repeats we have, that what tells what you look like. So you guys are doing your dog breeds right now, and you know that there is a huge diversity in the way that dogs look. And they're the only animal like this on the planet. There's no other animal on the planet that has this slippery genetic slope to their DNA like dogs. It's only 0.1% different. 99.9% of a dog's DNA, no matter if it's a Chihuahua or a Great Dane, it's only 0.1% of these repeats that gives the Great Dane its size, that gives the English Bulldog brachial-cephalic, makes the Borzoi dolichocephalic, makes the Labrador genetically prone to going to water, the Newfoundland also prone to water, the herding dogs, your border collies, your cattle dogs, that's what makes them herd, is this 0.1% that we alter the repeats and are able to manipulate shape and personality. In humans, obviously, we don't have that same diversity. I mean, our heads are about the same, and our features are about the same, and everything about us is pretty much the same. There's very little difference in our humans. but there are some personality traits that are passed down genetically. Artistic ability, scientific ability, some personality disorders like schizophrenia and OCD, even alcoholism can be a genetic repeat in the DNA. I find that all really fascinating. So when you were born, it was already determined if you were going to be a certain height, even a certain weight, depending on how your metabolism worked. What color were your eyes going to be? Oh. What color was your hair going to be? What color was your skin going to be? Were you going to have some sort of OCD? Were you going to be prone to arts, music, drawing? Or were you going to be prone more to the sciences, math, and biology? All of that comes from these 4 DNA nucleotides. 4 proteins make up literally everything about you. I find that really fascinating. RNA is how DNA sends messages outside of the nucleus. So DNA lives in the ivory tower of the nucleus. And DNA is not supposed to leave that nucleus. But he does need to send messages. We need to make things. We need to do stuff. And so he uses this RNA to send messages, to create other proteins. And it's going to transfer these instructions out of the nucleus. So these nucleotides, they are,
for a second?
of course I can go back. Absolutely. No worries. In fact, while you guys are furiously writing, I am going to go blow my nose real quick and see if I can get the sneeze out of me, because now it's starting to hurt. All right, that thing got it. Okay, so let's talk about these nucleotides. They are, as I said, they are sugars and proteins. They are a pretty complex makeup. And in DNA, we have the sugar portion called deoxyribose. And then in the RNA is just ribose. And that's hence the letters deoxyribose nucleic acid. And in RNA, it's just ribose nucleic acid. So those are how we get the letters DNA and RNA. And as I said, they're named. I'm not going to make you reproduce this. This is just a picture for those of you that want a little bit more of a complex understanding. I don't. So we have these 4 nucleotides found specifically in DNA: adenine, guanine, cytosine, thymine. 2 of these bind together and the other 2 bind together in DNA. And this is how I remember it: adenine and thymine. Those 2 bond together. Guanine and cytosine. These 2 bond together. Adenine will only ever bond with thiamine, and thiamine will only ever bind with adenine. Same thing with guanine and cytosine. How do I remember that? Apple tree, put your car in the garage. I know it's kind of a nonsense sentence. Apple tree, put your car in the garage. But that's what helps me remember. It's very simple. Apple tree reminds me what adenine and thiamine, they bond together. Put your car in the garage. Cytosine and guanine bind together. So apple tree, put your car in the garage. That will help you remember in DNA which binds with which. Now, this part is important. And I am going to test you on it. I am even going to tell you how the question will be structured. Which of the following nucleotides is only found in RNA? In RNA, thiamine is replaced by this guy, uracil. So tell me, now RNA, that's actually, I'm not going to say tell me. RNA is half of a DNA. Basically what happens is DNA has the blueprints for literally every tiny speck about you. And so your body says, hey, DNA, we're getting low on this certain kind of protein. We're getting low on this certain kind of thing. DNA has the recipe for that because he's the one that is the creator. And so DNA can go through and find the exact area in himself that is the thing that you need to make. It's basically like him, breaking out a giant book and turning to the appropriate page. He can then make a copy of that page, send the page out to the factory, and have the factory manufacture whatever it is your body needs. That's kind of the magic behind DNA. He can copy and replicate himself and send instructions to other areas of your body. And when he's copied these instructions and he has sent them out to the factory, he's not sending himself. He can't break off a piece of DNA. What he's doing is he's copying half, and that half is known as RNA. So it's really pretty interesting how all of this, and every single cell that has a nucleus in your body is doing this without us even being aware. The stuff that our bodies do without us even being aware of it is astronomical. That our bodies are on autopilot, taking care, making sure we're alive, more than anything than our conscious thought can do. Our conscious thought is such a tiny percentage of anything our body does. It's ridiculous. We really are like kind of automatrons. Our bodies just function independently and do what it needs to do when it needs to do it. I find that a little terrifying, honestly, that I have that little control over the functions of me. That's kind of crazy. Now I'm freaking myself out.
I have a question.
Yeah.
So DNA, I guess, what I got out of this is that DNA provides the instructions and RNA carries that out. Is that correct or no?
I'm looking up because I've put you guys up on my secondary monitor, so I'm not staring at my ceiling. I'm staring at my other monitor. So, essentially, now, have we simplified DNA and RNA to the nth degree? Of course we have. But yes, essentially, DNA contains the instructions. It's a great big recipe book. And then he copies down the specific recipe and he sends it out with as RNA, and RNA is going to go and take it to the factory, which is a ribosome, and the ribosome will manufacture whatever it is we need to manufacture. Does anyone else have any questions? Connie, I love that you have an alien behind you. I love him. I love him. See, on my monitor now I can see everybody. Almost everybody. Yeah. There you are, everybody. Tiana, I love the bow in your hair. Very cute. Tiana's got the most,
Thank you.
the prettiest pink bow in her hair. Yeah, you guys forget that I can see you, right? All right. So this is just basically a slide outlining everything I just said. These nucleic acids, these are going to just form great big long chains with their base, and they're going to have the information needed to produce proteins. They are the instruction manual to produce proteins in your body. And that's going to be based on the order of the adenine, thymine, guanine, and cytosine. Depending on the order and if they start repeating, that will determine the recipe for the protein. And that's it. That's all I need you to know. Oh, I did get ahead of myself though, didn't I? So we have these bonds between adenine and thymine, apple tree, cytosine and guanine, put your car in the garage. And basically, we can rebuild DNA. We can rebuild structures with only RNA. Let's do it. Here, I'm going to build an RNA. And we're going to see if we can, from this RNA, which is only half, we unzip part of DNA, which is the instructions. We're going to see if we can recreate what the DNA looked like. So I'm going to put here my nucleotides. So remember, RNA is only half. So these are all of my nucleotides. So this is cytosine, adenine, guanine, thymine. We'll do another thymine, cytosine, adenine, guanine. And so this particular RNA is sending out the recipe for a certain kind of protein that we need in the body. I don't know what this is. I'm just pulling something out of the hair. If I want to reassemble this particular DNA, can I do it? So what has to go here on this particular rung of the ladder? If this is cytosine, what does this have to be?
Guanine.
Yeah, this has to be guanine. And if this one is adenine, what does this one have to be?
Thymine.
Nice. And you see how now we can just fill in the blanks. Guanine, this has to be cytosine. Thymine has to be adenine. Another thymine is adenine. Guanine, thymine, cytosine. I have now just replicated a specific part of DNA. That's amazing. That's pretty incredible. And then as we are creating this and these nucleotides are coming together, that's where we're starting to get that spin, that double helix that we so associate with the DNA as it starts to twist around each other. And so the order of these nucleotides is what makes this genetic code individual. It's what gave you the shape of your nose and the color of your eyes and the shape of your eyes. It's also what gives you some traits that look like your mom, some that look like your dad, and some that could be from grandparents, great grandparents, because you're going to get repeats and you're going to get combinations that are contributed from every single person in the history of evolution that has contributed to your genetic makeup. There has never been a genetic makeup quite like yours. We'll talk about identical twins here in a second, but if you're not an identical twin, there is no genetic makeup quite like yours, and there never will be again, ever, ever, ever. You are a one and done. You are going to be a flashpoint in the pot, and when you pass away, when you die, your DNA sequence dies with you. It will never again in the history of man be repeated ever. Identical twins, absolutely I can. If I'm going too fast, do you guys need me to go back a little bit? Do you want me to go back to the previous slide?
I have another question.
Sure.
Are we going to be matching the nucleotides like on the test? I guess like you're drawing.
I think I have a question, and I know my drawing is not good. Here's what I can do. Let's practice that again. But this time I can. So I am going to
So we should know which letter like corresponds with.
Yeah, you need to know which corresponds with which. Apple tree, put your car in the garage. It
Okay.
really doesn't matter how long I make this. As long as you remember apple tree, put your car in the garage, you can replicate any DNA at all, ever. You can replicate an entire strand of infinite DNA. So here I have part of a DNA, okay? I have one tiny infinitesimal structure of half of DNA. And I'm gonna make a little bridge like this. Okay, so now the first nucleotide I have in my pretend DNA is adenine. Tell me what has to go on the other side.
Thymine?
Yeah, apple tree. Here I have thiamine. So I'm looking at the top, the ones that are given. What is automatically going to have to bind with thiamine?
Adenine?
Adenine. Okay, here I have guanine, G. Who binds with G?
Cytosine?
Nice. There's another G, so I know that's a C. All right, here I have a C. What binds with C?
Guanine?
Yep. Here I have another A. What binds with A? Adenine.
Thymine,
Yep. Just yell them on out. There I got a thiamine. What binds with thiamine?
Adenine?
Yep. Here's another guanine. So what binds with it? Yell it out.
Cytosine?
Cytosine. Good. Here I have a cytosine. Going to be guanine. So I can only match one with one. Adenine will only ever bind with thiamine, and thiamine will only ever bind with adenine. Apple tree. Cytosine will only ever bind with guanine, and guanine will only ever bind with cytosine. Car in the garage. And that way I can actually take half of a DNA and replicate it perfectly. So I do want you to know, and so I might have like a matching question where I would say, okay, which of the following combinations is correct to match with this half of a DNA? And I would give you this, not this long, I would give you maybe 3 or 4, but I would give you this and you would have to select the one that looked like this. Is everybody on the bus with this? Did anyone fall off the bus? Does that answer your question, Vanessa?
Yes, thank you.
Absolutely. I know it's hard to draw on the screen sometimes. So I need to remember that I do have this whiteboard at my disposal.
Yeah, kind of just seeing how each connects to the next one definitely helps.
Because, yeah, that's what I was trying to do with this mess up here. But it's really hard to write on this. All right, so let's play again. So this light blue is 1/2 of our DNA, and the dark blue is the other half. And it has this windy, twisty look because of the way it's shaped when we have these nucleotides binding with each other. Therefore, here I have, let's just assign letters to these. So let's say, Stylet is not working today. Let's say this is A. This one's T. Let's say, okay, well, that one we
In the corner of the picture.
do what?
There's like a key for the colors.
Oh, there is a key for the color. I didn't even see that. Ha! So there is a key for the colors, right? Come on. Right here. There we go. So seeing the keys for the colors, okay, we're looking at dark red. This is going to be cytosine. So what do I know this one has to be? There's even a cheat key right here.
Guanine.
Yes. If this light white one, I don't know what color that is, kind of a peach, is thiamine, what does this yellow one have to be?
Adenine.
Adenine. Apple tree, put your car in the garage. And so what this is showing you is just simply, I can have half of a DNA. I can unravel half of the instructions and send that out and then reassemble it perfectly. Because there's only one way to reassemble it. It's like IKEA furniture. There's only one way to put it together. And if you don't put it together that way, it's not going to work and it's going to look terrible. I am the queen of IKEA furniture. I love IKEA. My guest room, not for my room, but my guest room, I've got all IKEA because my about 5 years ago, my significant other's son and his brand new girlfriend, who is now his wife, she's adorable. I love her to death. They were coming to Texas. I was meeting her for the first time. So I only had one guest room that had a little single bed in it. Well, that's not going to work. So I had to redo the entire, I did. I spent the money and redid the entire guest room for them to come over. And it's all IKEA. And it's just like DNA. You have to put the right screw into the right hole or it's not going to work and they're going to fall through the bed. All right, so now we're going to talk a little bit about RNA. RNA, remember, is just the instructions that we're sending out. Okay, DNA has gotten the message. We need to send something out. We need to build something within the body. RNA is where DNA is going to unzip that part of the instruction manual, and it's that copy that's going to get sent out and create the protein. So we've got 3 forms of RNA, and I like to give them personalities. Of course I do. Christian, I'm not sure what you're talking about with nucleic acid formation stuff. We've just spent the last 30 minutes talking about how adenine and thymine bind together, cytosine and guanine. I need you to clarify your question just a little bit. No. So this is just nucleic acid formation. This was just going into that, this is what's making up all of us, the genes, and it's the information needed to produce proteins. That's all we went over on the slide. All right, so these 3 forms of RNA. So I've got my DNA up in the ivory tower. There's my poor attempt of a double helix. And he gets the message, hey, we need blah, blah, blah protein. And so what he does is he has to copy down. I imagine him having this massive book of everything that is you. And he looks up the name of whatever protein, whatever thing he needs to create, and he copies down the recipe, right? He gets a little piece of paper and he copies down that recipe for whatever it is we need to make. He's transferring the recipe to this piece of paper. No different than if you're like, oh, Ms. Huff, I think your lasagna recipe sounds amazing. Can I get some? Can I get the recipe? I'm going to copy the recipe for my lasagna down on a piece of paper. I have transferred the recipe from one piece of paper to another. This is transfer RNA. Then what I'm going to do is I am going to give this piece of paper to a messenger, and it's going to deliver it to the factory. The messenger is going to be messenger RNA. And then when it gets to the factory being a ribosome, the factory opens this up, reads the recipe, creates the thing. That's ribosomal RNA. So these 3 terms are literally just saying what is happening to the RNA at that moment. Am I transferring the instructions? Transfer RNA. Am I shipping the instructions out of the nucleus to the factory? Messenger RNA. I really do have this mental image of this little, RNA strand on a bicycle, like a bicycle messenger, delivering it to the ribosomal RNA. So that's going to be the factory, the ribosome of the cell. And then the ribosome is going to take that instruction manual and it's able to pluck the free amino acids and assemble whatever protein DNA has told it to assemble. Those are the 3 forms of our RNA. Do I have any questions about any of these 3 forms? I'm going to transfer, I'm going to copy it down, and then I'm going to messenger it over to the factory, and then the factory, the ribosome, is going to build whatever it is I've asked it to build. Any comments, questions, concerns?
Question.
Yes.
Do these 3 different types of RNAs look similar or different?
So they are going to look the same. And basically what we're talking about, at least simplicity. Right now we're just dealing with simplicity. Simplistically, these 3 right here would be 3 steps to creating one protein. It's just saying, where is it in the process? Have I transferred the recipe? Have I copied down the recipe? Has that recipe been messengered out of the nucleus? And has it been received by the foreman of the factory? So these 3 are in fact the same RNA in our minds right now. These are just 3 different steps in the process, and it's telling us where in the process are we as far as the creation of that new protein. Does that make sense?
Yes, thank you.
Absolutely. So here for you little visual learners, here's my DNA. And here I can unzip myself. This is my copy. This is my copy of this DNA. And so I've copied it. Therefore, right now it is transfer RNA. I'm going to ship it out of the nucleus, messenger RNA. And I'm going to send it to the factory, the ribosome, ribosomal RNA. And that's just going through the stages. And then ribosomal is going to read the instructions and make a protein based on what it's being told. Here's another little picture for you. Here's my DNA, and it's received a message. Uh-oh, we're getting low on this one thing right here. It's going to unzip itself. It makes a copy of the instruction manual. So here we are transcribing it. Here we're moving it out of the nucleus. Now we have our mRNA where we're actively moving out of the nucleus. And these little guys are my ribosomes. I take it to them. They read the instructions and you can see bloop, 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 bloop. They actively make the protein. So we have our transcribed DNA, messenger moving on out, and our ribosomes actively making whatever the ingredients list tells us to make. We're going to talk a lot more about this DNA unzipping and rezipping when we get into a further chapter. For right now, we just need to know, here's the same thing, it's just a picture, we're unzipped. Here we're moving it on out, and here we've delivered it to the factory or the ribosomes that read it and produce whatever it is we've told them to produce. All right, ATP, let's talk about it. You've heard these letters before if you've taken even the most basic of biology courses. You know that ATP, adenosine triphosphate, provides the energy for the cells. So how does it do that? Well, I have 3-phosphate, triphosphate. I have 3 phosphorus molecules bonded together. And what happens inside my cell is, I break one of the bonds. What is released when I break a bond?
Energy.
Energy. I'm going to release energy. And that little lightning bolt, oops, that's my eraser. My little lightning bolt of energy is going to be what provides energy for the cell. And so now I have diphosphate. Then I'm going to come back and I'm going to reform a bond and convert di to tri, break the bond. And it's just simply breaking, reforming, and breaking and reforming and breaking and reforming that ends up giving us the cellular energy that we need. These very high energy bonds are broken, energy is released. And so this adenosine triphosphate molecule, these 1, 2, 3 phosphate molecules joined together, these are what create the energy for the cell. So right now, we'll talk again. We're going to talk a little bit more about this in our next chapter when we get into the inner workings of the cell. But for right now, you just need to know adenosine triphosphate is 3 phosphate molecules bonded together. We break the bond. It's very high energy bond that is released, and the energy is released, and that gives our cell energy. So it's the conversion of ATP to ADP, and back again, back and forth and back and forth and back and forth. Does anyone have any questions about that? See you guys furiously writing. Hey, what are you guys doing in, I need to know exactly where you are in medical terminology right now. What are you starting on Monday?
We just finished the colors. Then we're gonna start lectures
You just finished what in MedTerm?
with the colors.
The colors. Okay. What chapter are you starting on Monday?
I'm not sure what chapter we're starting, but we just finished doing restraint techniques for dogs.
I think she said it was gonna
No,
be the
in
skeletal system.
the
Yes,
skeletal system in MedTerm?
I think it's chapter 5.
Okay. Good, good. All right, so ADP, ATP, why do we care? Why is any of this possibly important? Well, it's important because when we get further on into the medicine part of veterinary medicine, we need to know this information because of certain drugs. Certain drugs reacting within the body, certain drugs will speed up the rate that these phosphorus molecules are being, having their bonds break, providing more energy. Some of these drugs will inhibit the phosphorus from breaking bonds and therefore decreasing the amount of energy. These drugs can prevent or speed up different chemical reactions within the body. And so when you guys get into pharmacology, which is the study of medications, if you have at least a basic fundamental understanding of the conversion of ATP to ADP and back and forth and back and forth, it's going to be a lot easier for you to be able to understand the pharmacokinetics, or how do drugs work within your body. See, I'm using MedTerm now. Pharmacokinetics, the movement of drugs in your body. Kinetic is movement, pharmaco is medication. And so it's this energy, either lack of energy or increase of energy, that does allow your body to move drugs throughout it. So it's also going to help us when we start talking about digestion. Next term, when we get into the systems and anatomy and we get into digestion and nutrition, we're going to learn how does the body break down these nutrients, and how do we have cells talking to each other and signaling each other, and how do we transmit nerve impulses, which is just packets of information to and from my brain. All of this that we've just talked about, the DNA, the replication, the creation of new proteins and new substances, the messenger going to the factory, releasing energy by breaking the bond of the phosphorus and the triphosphate. All of that is going to be so important as foundational basement concepts that are going to to help you through. And no worries, Vanessa, I'm always interested in what you guys are doing in intro. I might have to take over for a few lessons in the second period midterm class. And so I just wanted to find out where exactly you guys were in midterm. And I'm happy to hear that you guys are doing restraint. That's awesome. Have you gotten to do anything on live animals yet? Okay, it's coming. We'll be bringing in some animals for you to do that too. Usually Miss Cat brings her dogs in. They're pretty cute.
I checked the midterm Moodle, and the next like PowerPoint she has is the skeletal system.
Perfect. Thank you, Avery. All right, so that concludes this kind of introduction PowerPoint. Now, I want to show you something that I've done for you. Now, we're not doing the test this weekend. Probably not going to do the test until Tuesday. That will give us some time on Monday to do some review. But I want to show you something that I've done for you. If you go to my website, not the Vet Tech Institute website, my website, and you look at the column all the way to the right-hand side. Oops, what am I trying to do here? You're going to see a PowerPoint that is called Chapter 2 Testable Material. Because I know that Chapter 2 does have a pretty substantial amount of information. So what I've done is I've taken the PowerPoint and I've highlighted everything that is potential test material. So anything that is in red is fair game for the test. So here, I do want to point out though, if something like a title, oops, if something like a title, like here, I just have, oh, it's not going to let me do it. I just have right here, oh, there's a laser pointer. I just have major elements highlighted. What that means is you need to know everything in underneath that title. So this is going to take, I've also taken out any slide that did not have anything to add to the test. So I have taken this 70 some odd slide PowerPoint and it's now like 35. So this will help give you, it's on my website on the anatomy tab, all the way on the column on the right-hand side, and it says Chapter 2 Testable Material. And so I've taken out any slides that didn't apply. I've highlighted the material that could be testable material, and I've taken that, 78 whatever slide PowerPoint, and I've reduced it by almost half. So hopefully that will kind of funnel and direct what you're going to be responsible for the next test. And then on Monday, I'll give you some examples of some test questions and you know, some wording, and give you an idea of what to expect on the test next week. All right, do I have any comments, questions, concerns? All right, so on Monday, we're going to get started with the next chapter, which is the amazing cell. And the majority of the amazing cell is going to be review. It's going to just simply, everyone knows mitochondria is the powerhouse of the cell. So we're going to be going over the parts of the cell, the function of those parts, and we're going to pick it up. The speed is going to pick up just a little bit. I'm going to make sure everyone's on the bus, but we do have a little bit of ground to cover. So we're going to be starting that on Monday. So if everybody will kind of read over the beginning of Chapter 2, kind of look over the beginning, or not Chapter 2, Chapter 3, kind of look over the beginning of the PowerPoint so that we are all starting a little bit ahead of the curve. That would be great. And if there's no other questions,